Welcome to Clean Lakes Alliance's Yahara Lakes 101 Lake Science Cafe. And I'd like to share just this right off the bat, and we talked just a little bit earlier. This is the largest group we've ever had at one of these meetings. So for getting up this morning and getting going and caring about the lakes, give yourselves a hand for me if you would, please. My name is Bob Weber. I'm president of First Weber, and also a member of the Clean Lakes Alliance Community Board. And I'd also like to just acknowledge Marilee Gorman. Are you in the room? Marilee also is one of my comrades at First Weber and a long-standing supporter of the Lakes and Clean Lakes Alliance and all its factions. So thank you, Marilee, for your work. And I'd like to also share, along with the hosting sponsor, the Edgewater, the University of Wisconsin Nelson Institute for Environmental Studies, and media partner, the Isthmus, and supporting sponsor, Johnson Bank. First Weber is proud, once again, to be the presenting sponsor of Yahara Lakes 101 Lake Science Cafe. Today, we're going to talk about a very timely topic. It's the blue-green algae blooms. But before we get there, we want to recognize businesses and organizations that became lake partners in June. They're joining a growing list of over 140 businesses and organizations that are supporting our lakes. Additionally, as well with that, I'd also like to jump to a very successful Loop the Lake bike ride, which was last month. We had over 900 riders participating, which is 300 more than what we had last year. So really, really a strong group that we had out there last month. And beside the events like Loop the Lake, Clean Lakes Alliance is working hard in the community to improve our lakes. Over 70 citizen monitors around all 5 lakes are the first to report the lake Cyanobacteria, or commonly called blue-green algae blooms, that have closed our beaches over the past several weeks. This data, which is available on lakeforecast.org, is truly the first line of defense alerting the public to these harmful bacteria that affect our waters. Also, our Environmental Impact and Policy Committee is working on an advocacy agenda in the next few weeks to recommend how the public can continue to push for action towards funding Yahara Clean Engineering Report. Clean Lakes Alliance is also looking at recommendations from the Healthy Farms, Healthy Lakes Task Force, which is comprised of farmers, county representatives, Clean Lakes Alliance Executive Director, and members of our community board. In the coming months, look for Clean Lakes Alliance to continue to push for more programs, more monitoring, and more phosphorus reduction projects. And with that, please join me in welcoming Kevin Ripp from Aquafix, who will tell us more about today's speaker and topic. Kevin.
Hi, I'm Kevin. I'm with Aquafix, or we have another division called National Lake Biosciences. We get in samples of algae and aquatic weeds from around the country, and we grow it to try and figure out how to prevent it. We try to figure out what drives it. So we study the levels of all the minerals in the water. We study the microbiota on the algae, and then in time we develop products naturally to take care of it. But we're very proud to be a part of Clean Lakes. It's a great organization. So Trina today is going to be talking what I call the 8th wonder of the world. And she'll be talking about how capable this cyanobacteria is. It's been around for billions of years, and it's got a lot of advanced processes that it can do, especially if you try and kill it. But it's been around for a couple $1 billion a year, a couple billion years. It's responsible for putting oxygen in the atmosphere. There's a whole bunch of things. And Trina, civil and environmental engineering, bacteriology professor at UW Madison. She studied at Berkeley. Her interests include microbial ecology, water quality, environmental technology. She's authored over 70 articles in her graduate program, and she has a group called the McMahon Group. Anyway, I want to welcome Trina.
Can everybody hear me? Can you hear me in the back? Good. I tend to walk around a lot, so I'm going to use this thing. Hopefully hopefully it works. So thanks everybody for coming out. I'm a little bit intimidated. This is even a bigger group than I normally teach at UW, so I'm nervous. So thanks for inviting me. I'm a professor in civil and environmental engineering and also in bacteriology at UW, so I am trained as an engineer, but I heard the siren song of microorganisms when I was a PhD student and started studying the microbiology of wastewater treatment. And when I came to Madison, I looked around and I thought, there's all these beautiful lakes here. There are world experts in studying these lakes in the Center for Limnology, and I said, I have to get into this. So I learned everything I could about the microbes that live in our lakes. And of course, one of the things I had to learn about was cyanobacteria, or blue-green algae. And so I had to sort of get a second PhD in understanding the organisms that are affecting our lakes. And I want to start out by asking you a question. And so, I make my students speak up, so I'm expecting you all to raise your hands and you can shout it out if you want. Is it safe to go in Lake Mendota today?
Yes.
Yes. How do you know? One person
It looks okay.
It looks okay, all right. What else? Any other?
Lake forecast.
Lakeforecast.org, right? Yay, Clean Lakes Alliance, good job. What else? Is there any? People aren't getting sick, but we know. I'm not trying to scare you, I promise, but. And I'm not trying to undermine the excellent work of CLA and of the Public Health Department because they do amazing work. And if anything, they're a bit conservative in when they tell you that the lake is not safe to swim in or boat in. But for me, that's a bit of a problem because that conservativeness actually prevents people from using the lake when they could be using the lake, right? And we know that if people get this perception that the lakes are dirty, and there's no way to clean them up, and we should just give up on them because it's too expensive to try to clean them up, then people aren't going to be invested in doing the things that we know will help the lakes, right? So if we can get a better handle on when it's actually safe to go on the lakes and when it's not, then we might have more people using the lakes, and that might lead to better outcomes in terms of people's investment in policies that will help actually help the lakes get more clean. So that's one of the things that motivates what I do. And so if we have this problem of not really knowing for sure if the lake is safe from the perspective of blue-green algae, I always get the question, why can't we just measure the stuff that makes it unsafe? Well, if you really, really want to know if these chemicals are in the water that the blue-green algae are making that would potentially make you sick, if you take that water sample and you go to a lab that will test it, it's about $250 a sample. So if you want to go and sample all the beaches every other day or every day, that's a lot of money that nobody wants to spend on that, right? So the turnaround time is also slow, so just based on the processing that has to happen with the sample. So by the time you get the information, the lake might be in a different condition than it was when you collected the sample. And then you also have this limitation just in the number of samples that can be processed. There's only a handful of labs in Wisconsin that have the equipment needed for that. One is the State Lab of Hygiene. One is at the University of Wisconsin-Milwaukee in their public health department. So there's limitations for what we can do in terms of getting that real-time information to tell you more accurately whether or not the water is safe. So I want to have a little bit of a bone to pick here, so you have to forgive me in terms of introducing some jargon. So we use the term blue-green algae because it's what people recognize. The water is bluish-greenish when they're present. They do kind of look like algae, but they are not algae. They are what we call cyanobacteria. They have a lot of the same features as algae. In fact, one of the theories that people have about how life evolved is that cyanobacteria were engulfed by another organism and that the organelles of plants and algae are actually the descendants of cyanobacteria many billions of years ago. So they're very similar, but cyanobacteria are also very special. They have different biology, different physiology, and so if you confuse them with algae, you can make some mistakes in terms of understanding why they are there or what they could possibly do. So that's just a nomenclature thing. I'm going to use cyanobacteria throughout my talk, but just so you know, it's a synonym for blue-green algae, but a little bit more technically accurate if you care about those things. So I wanted to talk today about things that we know and things that we don't know, because as a researcher, I'm always trying to figure out what, fill these knowledge gaps that would potentially help us solve the problem, right? And so I want to show you sort of the limit of what we know and why that might be influencing how we use the lakes and how we can clean up the lakes. So we know that if you put more phosphorus in the lake, you get more cyanobacteria. That I'm not debating. There's many people who have shown that, not just in our lakes, but around the world. We know that there's plenty of phosphorus in the Yahara lakes to fuel a lot of cyanobacterial blooms. So that's not up for debate. I have some ideas about other things that could be controlling their or driving their blooms beyond phosphorus, so that's what I'm going to talk about later. We know that some of them can make toxic compounds. So this is the concern about the safety, right? That if you drink the water, if you let your pet drink the water, that there's poison in the water that will make you sick. In worst case scenario, it will kill you because the toxins can be liver toxins, neurotoxins, things that really mess up your body. And there's not a lot of examples of humans dying, but there are plenty of examples of dogs that swim in the water, and humans that feel the effects of the toxins but don't die. And those are very hard to take a census of. The epidemiology of blue-green algae cyanobacteria exposure is a hard thing to get a handle on, because if you go to the doctor and say, I feel kind of achy and my head hurts and I have a rash, the doctor is she's not gonna say like, were you in the lake yesterday, right? Because that's not the first thing that comes to their mind. They're not trained to think that way. So we actually don't know how much exposure is happening and how much people are getting sick. So that's a challenge in and of itself. The other tricky thing is that you might have cyanobacteria in the lake that can make these toxins, but aren't. And we don't know why. We don't know what the switch is that turns them on, right? So you might have a really hideous bloom of cyanobacteria and it might not be toxic. You don't know, right? So, and measuring the toxins costs $250 a sample, right? So this is the problem. The other thing that people don't realize is that the toxins are actually, they stay inside the cells of the cyanobacteria. So they're not technically in the water until they die. So when the bloom is what we call senescing, when it's like rising to the surface and making that scum and looking and smelling really bad, that's when the toxins are the highest in their concentration in the water. Probably doesn't matter too much because if you drink the water, whether or not the cyanobacteria are alive, you're going to get the toxin one way or another because they burst inside your body or your dog's body. So that's a nice thing so the toxin levels are difficult to measure, but when we've done regular analyses of the lake, and we do have the funding to do that, we find them. We find different toxins at different times. We find different levels. Sometimes the levels exceed what the World Health Organization recommends as a drinking water standard. Sometimes they exceed the level for recreational standard, which is 10 times higher. So we have seen these sort of spikes of toxin concentration, at least in Lake Mendota. But it's something, again, we don't have a handle on because we can't sample frequently enough or in spatial enough resolution, like at the beaches versus the middle of the lake. So let's just talk a little bit about what I just went through, but from a more, let's give it a little bit more detail here. Again, we know it is the cow manure that is causing the density and the magnitude of the amount of cyanobacteria that is in the lake, right? That's not debatable. We know that precipitation at the wrong time, so rain at the wrong time of year, will put more phosphorus into the lake. So it's phosphorus that's in the cow manure that if it rains when the ground is frozen and there's manure on the farm field, it will go into the water that is running into the Yahara River and eventually into the lake, into Lake Mendota. So that's not really up for debate. There's lots of work, excellent work going on trying to control that. But given the fact that there's a lot of phosphorus already in the lake and that's going to take some time to figure out, we have to figure out what to do in the meantime, right? So more phosphorus equals more cyanobacteria. Let's talk a bit more about this problem of what do we know and what do we don't know about their ability to make the toxins. So these are my cyanobacteria. The Microcystis genus, so the flavor of cyanobacteria that I'm showing here really does sort of look like these little non-describable balls under the microscope. The red star is supposed to be a gene. So for those of genetics or that deep part of biology. It's a gene, right? But it's just the capability of that organism to make a toxin. So it's able to make toxins. So again, you might have a population, a group of cells that are in the lake where only a fraction of them are able to. Okay, so right there, the number of stars makes a difference probably, right? And we don't always know that. Not all of them are primed or able to make toxin at any given moment. So, you go out there and maybe a third of them are able to, only a third of those are actually doing the machinery, making the machinery in order to make that toxin. And again, we don't know. That's very difficult to predict. Now, and then there's only certain conditions when they actually can make the toxins, right? So you need to be able to, you need to be primed to, and then you actually do. And all three of those steps are things that we don't understand about the biology of the cyanobacteria, about the environmental conditions that would lead to the toxin production. So that really is the research frontier. So if you're in a lab and you're studying the cyanobacteria, those are the questions that you're asking. And so those are some of the things my lab has looked at, but then mostly in Wisconsin, it's Todd Miller at the University of Wisconsin-Milwaukee, who was a postdoc in my lab 10 years ago, who now is sort of the expert in the state. So if you have a question. Okay, yes. So it's possible, of course, that all of them are making the toxin. I guess I wanted to point that up in just a moment. When the cells die, toxins get into the water. Okay. So we just went through all the things in green. Hard to measure, but we find them when we look. So let's take a look at some measurements. And before I tell you the measurements, let me show you what we're actually measuring. So these are structures, chemical structures of the toxins that we worry the most about. There are others, there's a long list, but these are the really nasty ones. Like these are the ones like saxitoxin in very small amounts will kill you, and also cylindrospermopsin. Microcystin is the one that's most commonly found in lakes, in temperate lakes that have a lot of cyanobacteria and anatoxin. And the names of the toxins are sort of related to the names of the organisms that make them, but not completely. So the organisms can actually mix and match what toxins they make. So it's not always a good indicator of which organisms are making them. So Todd and some people in his lab did a nice survey in 2012 of the Madison area lakes, as well as also Fish Lake, which is a little bit north, and then Lake Winnebago, which is where Todd does a lot of his work. And so what I'm showing you here is a pretty complicated graph, although I have even more complicated than this. So the bars up here are different forms of microcystis. So if you march across here, these are different versions of microcystis. And then the bars are bigger or smaller based on the percentage of the samples that had a detection of that toxin. So it's not telling you anything about the level of the toxin or the amount that's in the water on that sampling day, but it tells you how frequently did we see it in the water above some threshold. And so you can see that, some of our lakes, you're detecting it all the time, or at least pretty frequently. There are a bunch of other, well, let me talk about these first. So here's those other really nasty ones, and so they aren't found very often, which should be, reassuring that the really, really nasty ones are infrequent, and probably we know it at relatively low levels. But there's this other category of compounds that Todd studies, which are not toxins that people normally recognize or that have been studied a lot. They're other chemicals that cyanobacteria make. We don't know why they make them. We think it's probably partly, talking to other organisms. They talk with their chemicals. It might be trying to poison other organisms. It might be just a process that they use when they're growing to sort of get rid of waste products. So they're similar compounds in structure and composition, but we don't know about their bioactivity. We don't know if they're toxic. Nobody's ever asked that question in a research context. So they could be making these other chemicals that could have effects that we don't understand. So that's an interesting thing to think about, that there's the toxins that we know and the toxins that we may not know. And that's one of the things that he's studying a lot. So the other thing you might notice is that if you're from Appleton or any other city on that's on Lake Winnebago, is that they have an even bigger problem than we do in terms of the amount of cyanobacteria and the toxins that they make, which is a concern because they use that lake as a drinking water source. We don't use Lake Mendota as our drinking water source. We use groundwater. But Lake Winnebago provides drinking water for a lot of people, you better make sure that your drinking water treatment plant is removing that material. And they do a good job of removing it, I should say. Here's an even uglier graph. So you don't need to think too much about this one if you don't want to. If you're a nerd like me, you can get into it. So each panel is a lake, and then these are different months across 2012. And what you should notice really is whether or not the data get above this dotted line, right? So if you see one of these points above the dotted line, that means the concentration of that chemical is higher than what the World Health Organization would recommend that you come in contact with, right? Or that really for drinking water samples. And so you can see that sometimes it is, but not all the time, and not terribly frequently in most cases. And these are, again, just the different forms of microcystin, and these are those weird chemicals we don't know much about, and those are the really nasty toxins. Microcystins tend to be the ones that are most abundant, most frequently detected. That's pretty common in a lot of lakes. And it's toxic, but again, not as toxic as some of the other ones. And I already talked about drinking water treatment in Lake Winnebago. So this is the kind of information that we get when we pay a lot of money, that $250 per sample. You actually get even more. There's like a long list of compounds that you get the information for, so you get a lot of data. But sometimes, even when you have those data, interpreting what it means in terms of the safety of the water is still a problem, because you don't know, is it, just because the World Health Organization set this number of 1 microgram per liter, if it's 1.1, is that a problem? If it's 0.9, is that a problem? So again, we don't, there's a lot of things we don't know about those levels that people think are safe or not. I guess I wanted to make the point next that that it's not just Madison that's struggling with this. We saw Lake Winnebago has a problem. This is becoming much more in the consciousness of people in other states and cities as well. Probably the one that gets the most press is the west basin of Lake Erie. And that's, again, because there's people in the western basin of Lake Erie that use it as their drinking water. So you can talk about Cleveland and Toledo. I think there was an episode a few years ago where they shut down the drinking water treatment plant for 500,000 people. Like, they couldn't use their drinking water, they had to get bottled water. So it's impacting a lot of people in the United States. Lake Okeechobee in Florida was in the news just yesterday because they're having really miserable problems. So it's not just us, and there's a lot of people thinking about this. Of course, the bigger lakes that provide more drinking water tend to get more attention and more money to study and try to solve the problem. I guess the other thing I wanted to point out is they're really beautiful, right? Like we get disgusted by them, but one of the reasons why I'm excited to study them is that they're really charismatic, especially at these big scales. So this is actually from remote sensing, so like from satellites that you can see. Even then, you can see the spatial distribution. This is a boat, right? So this is a zoom in of Lake Erie. So you see these structures that are forming because of the water movement, and so. We also don't understand how the cyanobacteria get transported long distances. You can notice on Lake Mendota even that there's spatial patterning, so that half of the lake could have a bloom and the other half does not, which again is a really hard thing to get a handle on when you're trying to predict whether this beach is safe and that beach is not safe. And that can change over a matter of hours based on the wind direction and the wind intensity. So that's another thing that we don't understand very well, but really brilliant people are thinking about it. And working towards trying to understand it better. So this is just another zoom out of Lake Erie and to make the point that they have a problem. This was 2017, last year. And I think they already are experiencing some pretty intense blooms this season. It's not just the United States. There's bigger problems in developing countries or sort of semi-developed countries. These places tend to have no regulation or very little regulation on where wastewater goes, so human sewage. This is Lake Taihu, which is in China. It provides drinking water for millions of people, like many millions. And so when they have blooms like this and they shut down the drinking water treatment plant, that's a big problem. And just the fact that people who use that lake for their food source, for many of them, that's their primary source of protein out of fish in the lake, that's a concern as well. And so it's not just us, but it can actually be worse. I would say that's a lot worse than Minnesota, actually. So So I want to go back to looking at the toxins. And so now I'm going to ask you to think a little bit about chemistry. So who in here has had an organic chemistry course? Awesome. So when you look at these compounds, what do you see? What features do they share? Or not share? So if there's, these mean that there's carbon, right? We implicitly, the way that we look at chemical structures, if there's a carbon, you don't write the letter, right? So this is a carbon, these are bonds between carbons, these are hydrogens, right? So on average, these compounds contain more nitrogen than what you might expect, right? Just based on sort of the stoichiometry or the relative levels of elements in the organism. So they're enriched in nitrogen, and so that's something that's always puzzled people, why that would be, why an organism would invest so much energy and effort into making these complex structures that actually use an element that they need to build their own cells, right? So they're wasting a very precious resource by making this stuff. And so, again, we don't understand why they make it. And so that is one of the puzzles that we have. But I have a hypothesis that I think sort of it starts to get towards explaining this, maybe not why they make it and why they're enriched in nitrogen, but why the compounds are made, or the chemicals are made when they are. So what I'm going to talk about is not, I guess, it's not something that anyone has proven or provided a lot of evidence for. We have some hints of it in some work that Todd Miller did in my lab and Lucas Beaversdorf did about 10 years ago. And so trying to put all that together and put it into a hypothesis, something that we would like to test, we'd love to do research on and try to figure it out. So I guess I wanted to preview that before I tell you what it is. That, don't go telling everybody that Trina has figured it out, because I haven't. Thanks, Nick. So cells in the lake often, have everything that they need. They have, the main things that a cell of cyanobacteria needs. It needs light to get energy, right? They do photosynthesis. If they're near the surface, that's not a problem. They have lots of it. They have lots of phosphorus. We already talked about that. It can be limiting sometimes, or they can run out of it sometimes, but on the whole, they have plenty of phosphorus. They have plenty of carbon dioxide because it's in the atmosphere, right? The carbon dioxide actually dissolves into the water, and then they can use that and they fix it. It's called fixing, right? It's just like a plant would, and turn it into their cell, into their biomass, into their cell components. So they have plenty of that. It's unlimited. It's the atmosphere. Nitrogen is a tricky one, though. So nitrogen is also in the atmosphere, but only certain organisms can get it out of the atmosphere and turn it into something useful. Otherwise, they have to use the nitrogen that's dissolved in the water in the form of nitrate or ammonia. So again, if you're a chemist, you can know the difference between those. If you don't, it's totally fine. You don't need it. But the thing I think is going on is that sometimes they run out of nitrogen at a really critical point in their life cycle. And let me tell you what I mean by this. So microcystis can be capable of making the toxin, right? That's what the red stars are. They can be primed or ready to make the toxin, but they can't make it because they don't have enough nitrogen. So they're limited by the nitrogen. It's been used up to make their cells, to make all the other cells at that moment in the lake, so it's tied up in the biomass of the organisms. And so there's another kind of organism that is frequently in the lake. It looks like grass clippings, but it's also a cyanobacteria called Phantomnon. It's just another genus of cyanobacteria. And these are the organisms that have this special capability of taking nitrogen out of the atmosphere and making it bioavailable. So they can fix nitrogen, and then they have unlimited amounts of nitrogen. So they can make enough for themselves, and then they excrete or spit out additional bioavailable nitrogen. Again, we don't know why, right? It doesn't make much sense why they would do that, but they do. And that nitrogen is now available for everybody else who's in the water, including the microcystis. And so what I think is going on is that as soon as they get that pulse, if the Phantomnon grows and secretes a lot of these nitrogen compounds, that the microcystis can get it and it's ready to go, and it makes the toxin. So what's going on here, I think, is not that nitrogen is necessarily limiting across the whole year or across a particular season, it's more that there's this moment when the cyanobacteria have grown that if they suddenly sort of become limited and then suddenly get it, that they can make more toxin. So it's a limitation that happens on daily to weekly timescales, which is very different from phosphorus, which is more on annual to seasonal timescale. The other weird thing is that we think athanasomnon makes toxins too, so that just confounds things more. They make their own flavor of toxins, but they can also make microcystin, which is the thing that microcystis makes. So again, complicated. So let's put this all together. So what do we know, what do we don't know? We know that cyanobacteria are limited by phosphorus in most temperate lakes, and that you add more to the lake, you get more cyanobacteria. That's not under debate. But the tricky thing is that we don't know when those cyanobacteria that are blooming are safe, right? Whether they can make a toxin, whether they or how much they're making, or even why they're doing it. And that's the research frontier. We think, or my lab thinks, and Todd Miller thinks this too, that there's something going on with nitrogen that we have to figure out. And that's an interesting thing to bring up. There's actually a debate among limnologists, people that study lakes, about the relative importance of phosphorus and nitrogen. If you start bringing it up, people get really upset. So if you, it's a war among different limnologists, like the academics get into this stuff. So like, oh, I think it's phosphorus. Oh, I think it's nitrogen. Oh, and you have these papers written and they're like criticizing each other. So I'm treading on dangerous territory to bring nitrogen, right? So I don't want to imply that we should switch all of our regulatory focus to nitrogen, because there's nitrogen in manure, there's nitrogen in other kinds of fertilizer. So I don't want to imply that we should put our attention there, because I think our attention should be on phosphorus, but that there's something going on there that we need to figure out, and that might be a clue to helping clean up the lakes, but maybe it's not. But in terms of understanding why and when there's toxin in the water, I think it's important. I guess the bottom line is, right now, if you're at a beach and the beach is supposedly open, but suddenly it starts to look bluish-green and you can see, what look like particles in the water, just don't go in it. Because right now we can't tell you for sure whether it's safe or not. If you see that the beach is closed because the public health department has run some tests, or Clean Lakes Alliance has determined that based on their citizen monitoring, just don't go, right? Because they have conservatively decided that it may not be safe, right? So there's different layers of being sure or not, and they tend to take the most conservative approach, which will, means that it might be fine, but you don't know, so just don't. Yeah, and I think that's all I have, so I'm excited to have your questions, and thank you for bringing it.
No questions on this topic. So Paul has a microphone here. We're going to try to move through as many of them as we can. We have a lot of time, so.
We'll start right up here in the front, and then just wave to me, raise your hand so I can see who has questions.
Hey, good morning, John Benson. Thank you, Trina, for a fantastic presentation, very educational. I keep small bodies of water in my home, aquariums, and I'm very interested in chemicals and how they impact the plant life and animal life that's thriving there. And I kind of have a God complex about how things work. So many of my dumb friends have the same fixation, but they scrub chemicals out of their marine and freshwater aquariums, like phosphate and phosphorus, and I resort to just photoperiod, plants, and proper balance. My question is, what strategies for phosphorus mitigation have failed that we know of?
Lean off to the hard one. Phosphorus is really hard to get out of the water, and it stays in the system, in the lake, because it doesn't have a form that is gaseous, right? So nitrogen can be N2, and 78% of what you're breathing is nitrogen gas. Carbon can be CO2, right? But the phosphorus doesn't ever go into the atmosphere, so it builds up in an ecosystem. And you can remove it with chemicals that will precipitate it, so turn it into a solid, which then it's heavy and it sinks to the bottom. So people have tried putting that chemical in the whole lake. You can use aluminum, so there's aluminum sulfate that you can put in the water, and it will make the phosphorus turn into a particle and settle out. And the idea is that it is buried in the sediment and is not available for the organisms in the water. There's issues with that. People don't like the idea of adding another chemical to their water. There's issues about aluminum and whether or not there's aluminum still in the water, and there's sulfate could cause a problem. So there's issues with that, but there are examples of places that have used that, and their lakes are crystal beautiful clean, right? So in Minnesota, they do this at sort of medium to small size lakes more often than we do. I think at the scale of the lakes in the Yahara River, that won't work because or in the Yahara Watershed, they're too big, right? And so it's only a certain size kind of lake that it works in. You've also seen people aerate the bottom. So the reason why phosphorus gets sort of recycled in the system is that you get the removal of oxygen at the bottom of the lake due to dead cyanobacteria, actually. And the phosphorus can then come out of the muck that's at the bottom. Just chemically, that's the condition when it comes out. And so people will pump air into the bottom of the lake. And that can help sort of, if you maintain it. So you have to do it all the time. It's not something that you fix and then walk away. In Lake Mendota, I don't think that will work because the chemistry of Lake Mendota is actually pretty unique in that there's other chemicals that are required for that process of release of the phosphorus to happen, and Lake Mendota doesn't operate that way. So I think even if we could afford to put a giant blower that puts oxygen in the bottom of Lake Mendota, that I don't think would work either. But really, I know one other thing people have tried. So related to that oxygen buildup, people will put nitrogen into the lake. So they will deliberately put a whole bunch of nitrates into the water, and there's sort of the scientific basis for that is actually sound to some extent, but then there's unintended consequences of adding the nitrogen, and again, that's only a short-term solution, and the phosphorus problem comes back. So there's examples. People have tried lots of different stuff. Some of it sort of works. Most of it kind of doesn't. Does that help? Yeah.
Are the toxins topically absorbed, or is it mostly an internal problem, GI?
It's mostly ingestion. When you drink the water intentionally or unintentionally, you get a rash from some of them if you come in contact with your skin. I don't think that we know whether those rashes, that irritation of the skin, is caused by the toxins that we normally look for, or those unidentified compounds that I mentioned, right? So that, again, the cyanobacteria are making lots of chemicals, and some of those might be the ones that cause the skin irritation and may not be the same as the ones that are poisoning you when you drink it. So that's something we don't know very much about.
So I really liked your correlation of nitrogen and toxin production thinking. So I know farmers really count on thunderstorms, lightning converting N2 to bioavailable nitrogen, and it's a lot. So has anyone correlated lightning storms with blooms that are toxic?
No, that's interesting.
That's an idea for you.
Yeah, that's very cool.
One of the problems of always studying something in 1 year, all that work in 2012 happened to be a severe drought year following 2011, and you showed some levels of toxins, but it wasn't a situation where we had a lot of nutrients in the water. You want to comment on that?
Yeah, that was unfortunate. The year they picked to do that really intensive survey was, well, depends on what you consider fortunate or not. The lake was really nice that year. Bummer. So we were out there in 2008 and 2009 and also 10 when it was pretty bad, right? So 2008 we had epic floods in June, and then so 2008 was bad. We almost had an echo of that in 2009, and then 10 a little bit. And the problem is we didn't have the technique or the technology for to measure all those different toxins back then. And so the ones that we did measure were a lot higher than what I showed. So they were up above to like 10 micrograms per liter of microcystin. So I didn't show those data because they're, we didn't have as sophisticated of a technique as we did 10 years ago that we do now. So high I don't know of anybody who's measuring, maybe Kirsty Sorsa or some of the folks from the State Lab of Hygiene can comment on actual toxin levels. Maybe it's something you don't want to talk about in the worst years, so especially last year and this year. So we have a stockpile of water that we have in our freezers going back to 2014 that we've been holding on to, hoping that the cost of the analysis will come down, or some, sugar daddy will come along and agree to do it for us. And Todd Miller, I've been talking to him for, since he left my lab, like, we've got to measure these things in the Yahara lakes. It's super interesting. He's always like, well, give me some money, Trina. But I talked to him 2 weeks ago, and he's so interested in the most recent blooms. He's like, send me everything you got, I'll run them for free. So I'm like, I need to get him to him before he changes his mind. But a few months from now, I'll have a lot more information about that, because we're bracketing the many years of drought. But you know better than I do, Dick, about whether, in 2014 to current, like how much variation there was in rainfall and bloom intensity. But my sense of just watching the lake is that, it's gotten a lot worse in the last 2 years, right? So we'll see that in our data. So stay tuned.
I'm going to get to the other side of the room next, but we have one more question.
Yeah, hi. My question is about dogs. So I had a dog a few years ago who was swimming in Lake Monona, and she got very ill suddenly, and she had, by the time we figured out what it was end-stage liver disease, and she died, and probably because of swimming in the lake, because she did that a lot. But my question is, one, I'm wondering if Clean Lakes Alliance could monitor the few dog beaches we have in Madison, and it's only like 2 or maybe 3.
Which ones are they?
Warner Park has one.
Warner Beach.
It's not it's not the regular beach. It's by the lagoon, but there is a Warner Park Dog Park. That's what it's called, Warner Park Dog Park. And then there's one Cherokee Marsh, a dog park, and they have set up for dogs with a beach and with a platform and all that. Those are the 2 main ones there are.
Governor Nelson.
Oh, Governor Nelson. Okay, other people have other ones too. Yeah, that's called, what is that dog park called? Cherokee Dog Park? Yeah. So just because I know, people in this hot weather like to take their dogs a lot, and now I'm so wary of it. So that was my first question.
It's a good one. I'm going to just
answer
quickly here.
No, but the other one is just related to dogs. So if they're not monitored, how do we know if it's safe for our dogs to go in?
So I think a good thing to start with too is I think we will want to look into dog beaches too. So we are monitoring all of the beaches and lake access points right now. But we do have a beach survey out. So cleanlakeslions.org/beach-survey. Is that right? Yes. If you go to the front page, cleanlakeslions.org, you'll find it. It's certain beaches. But we're trying to gather stuff like this right now. So we're doing this large gathering of information to find out what works, what doesn't, where us as a community needs to improve, this being one of those things. We'll gather all this data. We have some experts that are going to look at it, and then we'll have a state of the beaches that we'll release in May. But this is a prime example of something that we really need to look at as a community, because maybe it's not just us, it's our pet friends, and we constantly say if there's a cyanobacteria bloom, people and pets need to stay out of it. So I'd encourage you to take that survey and then come find me afterwards, and maybe we'll just kind of talk about some of those areas that maybe aren't monitored by our monitors, but if there's a way to increase it by people that use that, because all of our monitors are volunteers right now. So if somebody is using those dog parks on a regular basis, maybe we can tie that together. So that's my answer. I will.
So one other thing, we will send, if you registered at any time for today, or if you came in today and gave us a donation, thank you by the way, we will send out the link to you today. It's really important for us to find out, you as citizens, how you interact with the lakes, and what parks you go to, and what different things that you want. Because right now, I think our lake parks have an opportunity to be more eco-friendly, more sustainable, and more usable. When people use the lakes, then they're going to want to help the lakes. So we will send out that survey, and I do encourage you to take the challenge of going to one park this summer and rate that park of what you like, what you don't like, and what you would like to see happen in that park. We will then use that to advocate for changes for our parks to be more sustainable and lead the way to reduce phosphorus and runoff to the lakes. Got it? Are we going to do that? Raise your hand if you're going to do that. Thank you. Thank you.
All right, we got a question here.
Yes, I was curious, what happens when all of these massive blooms die? So do they go to heaven? Do they go to heaven?
Heaven or hell? Hell.
And then to the extent that they might have dropped to the bottom of the lakes, is there some kind of cumulative effect there? And does that lead over time to worse and worse blooms simply because we have all of this dead material at the bottom of our lakes?
Did someone plant you as a question? Things that I get most excited about. So, we don't actually know why a cyanobacterial cell would die. Like, that's an interesting, like, what, you know, they're just hanging out. They don't, you know, get cancer, or they don't like heart disease, or like what. Often it's a virus, so they get infected by a virus and they burst. But it's interesting that we don't really understand why. They might also just be accumulating damage from the sun and things like that. But when they die, you know, if they're heavy enough and they sink to the bottom, they're good food for other bacteria. So other bacteria, there's tons of bacteria in the water, right? Freaking out. Tons of bacteria. There are 10,000 bacteria in every milliliter of water. But maybe one of them will hurt you, right? So the thing is that there's lots of natural bacteria. We like to talk about the lake microbiome, right? Everybody has heard about your gut microbiome and how important it is for your health. So water has a microbiome. And so there are microorganisms there that aren't going to hurt you. They're just doing their thing, recycling stuff. If they weren't there, the lake would just pile up with dead things and wouldn't be a lake anymore. So they do their job of breaking down the dead cyanobacteria, but they like to breathe oxygen, similar, not sort of similar to the way we do. So they use up the oxygen, and so then the bottom of the lake becomes anoxic. That happens in Lake Mendota in sort of mid-June, so it's very early because there is so much rich material going down into the bottom of the lake. And that's when the phosphorus gets released again. All the other elements that are in the cyanobacteria get sort of recycled. And so as the summer goes on, the phosphorus concentrations in the bottom of the lake actually increase. It builds up and builds up. The nitrogen concentration, the bioavailable nitrogen, is also increasing over the summer. So when you get in the fall, like usually October, maybe mid to late October, when the lake turns over, so it mixes top to bottom, you get a huge redistribution of all those nutrients throughout the water column. So we actually get huge blooms in, end of October, but no one's paying attention because they're not driving their boats or, swimming at that time of year. But those are some of the worst blooms that we get, actually, because those nutrients are now completely recycled throughout. Some of the phosphorus could conceivably precipitate again, like into the muck at the bottom, but we think a lot of it's just getting recycled, which is why you can't get rid of it, right? It just cycles around and around. So, did I hit all the questions that you have?
So does it get worse over time?
It gets worse over time in the sense that you get more and more phosphorus, right? So phosphorus is in the system. It's in the cyanobacteria. Cyanobacteria die. It gets trapped at the bottom, mixes, and then it's trapped under the ice, right? And then in the spring, there's really high phosphorus concentrations that are left over from the previous fall, right? That gets fuel what happens in the spring, and so it's the only way it can leave is through the Yahara River going into Lake Monona, right? There is no other way for it to leave, right? So that's why it builds up, because it's often trapped down there. So for many months of the year, it's trapped at the bottom. So you're not getting removal.
I promise I'll get back up there, Adam. I promise I'll get back up there. I got 2 questions back here first.
We're coming up here then.
Which one of our lakes has proven to be the healthiest and why?
I can tell you what the nastiest. Isn't what Dick Wabisa's the worst? Wagra's the cleanest? Yeah. Of course, because you cleaned it up. I think there's too many weeds in Wagra. Dick? We don't know what to weed. Yeah, in terms of the algae, there's not a lot, not cyanobacteria. Yeah. It probably depends on what is clean to you, right? Yeah, healthiest in terms of the lake, or healthiest in terms of us interacting with, right? I don't even know the answer to that. I mean, it's probably is Wingra. even though, you look at it and it's kind of, there's a lot of weeds in it, there's a lot of plants. But that's healthy for the lake, and the plants don't make the toxic chemicals that the cyanobacteria do.
Can you talk a little bit about the life cycle of the toxins and how persistent they are?
That's also something that not a lot of people have looked at. And part of it is because to do it in a laboratory, which is the best way to get those accurate measurements, the water that you use in the laboratory might be different from the water, is probably different than the water in the lake. So you could bring the water in from the lake, but then you're already kind of changing the conditions. Most people who have looked at that, they find that it's on the order of hours to days, like maybe like 2 days max that it sticks around in the water. So it gets broken down by all those bacteria because it's got lots of nitrogen in it. It's super tasty for the bacteria that are naturally there. So they attack it, break it down, use it to build their own cells. So it's short-lived. But again, we don't know when that spike happens. And if we did do sort of a daily monitoring of a bloom a while ago, many years ago now, and it spiked like one day and then it was gone. So fast turnover.
If nitrogen is kind of like the elephant in the room that people don't want to talk about, is there anyone actually doing research that says, here's a small lake, we're going to control the amount of nitrogen that goes in terms of people's use of fertilizers on their lawns or what's happening in the farms around it, and see if that actually affects a lake? Because all the stuff that you buy to go on your lawn to make your lawn nice and green, a lot of it has nitrogen. And they talk about, add more, make it greener. Well, now I find out that might be as bad as the phosphorus that they took out of our fertilizer a while ago.
Mm-hmm. Yeah, well, in terms of fertilizer use, it's probably like find a good balance is probably the best advice. But I don't know of a study that's done what you just described, where they would take a lake that already has a lot of cyanobacteria, manipulate it, and measure the toxins, right? Again, because not very many people measure the toxins because it's expensive and hard to do. I'm sure there's been studies where people have just measured cyanobacterial biomass and seen if it's gone down or up. But I don't think that's the problem here. I think it's like, if you have the cyanobacteria, then the nitrogen becomes important. But there are people who are at a basic biology level working with, cyanobacteria in a laboratory and, subjecting them to different conditions and seeing, if we deprive them of nitrogen at just this right moment, how much toxin do they make. So there's people working on that. But how that translates to a lake in the field is harder. Not very many people can work at both those scales.
We've got 2 up here, and then we're going to go all the way to the back also. This one is
I have lots of questions on the toxins, but I'm going to limit my comment to just one issue. The waters and the pollutants that they carry, when they leave this area, they're actually going down into the Mississippi and contributing to the hypoxia zone down in the Gulf of Mexico. Is there a coordination with different universities and different agencies on looking at what's contributing to the hypoxia zone?
I don't know much about that. I know that scientists, there's lots of attention going to the dead zone. Ironically, I have a colleague at Louisiana State University who studies the same thing I do, and he's finding a lot of the same organisms that we see in our lakes, like in the coastal areas of Louisiana. But there's probably some level of coordination that's happening state by state, but interstate coordination is hard, right? For basically the way that our regulatory structures are set up in the United States, and it's certainly not as organized as like the Great Lakes Coalition that is partnered also with Canada. But that's about all I know. I actually shouldn't run my mouth too much about it because there may be something that I don't know about. Does anybody else know about coordination at the level of states along the Mississippi? Dick?
Well, there's a lot of national interest in where the nitrogen, say, in phosphorus is coming from. Wisconsin actually is relatively small contributor compared to Iowa and Illinois and the big corn states and things, so particularly on nitrogen loading. So yes, they do look at this at the very large Mississippi River Basin scale, where are the sources of pollutants? And not that Wisconsin doesn't have a contribution, but the Yahara Lakes, interestingly, the lower chain of lakes strip out most of the nitrogen, so what's leaving Kaganza actually is not that high relatively in nitrogen compared to what maybe you see up in the upper end of the watershed.
I'll adjust 4 minutes and maybe a question up here and then a question back.
Okay, we've had these two really massive cyanobacteria blooms in June, and my observations are just on Lake Mendota, and I've seen people recreating, kids down playing at the water's edge, people water skiing, too close, and so I've been writing to the health department and different agencies, and the World Health Organization has a alert at 4 micrograms per parts per billion. Is that correct?
Yeah, parts per billion is the same as microgram per liter, but
Okay.
yes.
Okay.
So one is the drinking water standard. 10 is a standard that I usually hear for recreation.
Okay, 10 is
four might be in some places. I'm not sure.
Okay, so 10 is the recreational.
That's the number I usually hear.
Okay, and so the Madison Health Department is what?
Well, Kirsty, 20 is 20 for recreation.
The 4 is the EPA draft, just limit for recreational water.
Right. So Kirsty works for the public health department. So
Yes, I've been writing to
I
her.
rely on her for the details, right? So yeah, 20 is what we use locally. 4 is what the EPA, so that's U.S. Recommended limit, right? Because again, it's hard. What do you enforce in that sense, right? So EPA can't enforce anything, right? They say we think you should stay out of the water if it's 4 or greater, but there's not a pressure point there. That you can regulate.
So my concern is, yes, we have lake forecast and we have wonderful communication from the public health department to the university, but still we are having all these people in there. Do you think, James Tye, our director, brought up at the last meeting that he thought that there should be a cyanobacteria alert the same way that we have pollen alerts, the same way that we have flood alerts. What do you think of that?
I think that's an interesting idea. I would love to have more data to drive that alert system, right? Because again, it's you definitely don't want kids and dogs playing in there if there's toxin, but you don't know if there's toxin. And if it's bluish, like it was in June, I agree. Like that was nasty. You shouldn't be in the lake. And so still people are. Yeah, I don't. But yeah, sometimes people, how is people getting their information?
I think information would be better if it's out.
Yeah, would those people notice like a pollen alert, right? So it's like, I think you need a social scientist to come in and be like, what's the best way to get that information to the people that need it?
We've got like 1 minute to fire out one more question here.
Hey, so my question has to do with sort of toxins and potentially entering the groundwater. Is there any sort of research that's being done on that, or are we concerned about that sort of thing?
Certainly not the cyanobacterial toxins. There may be other things that are entering the groundwater because there's a connectivity between the lake and our groundwater. And the flow has been reversed. So it used to be that the groundwater would feed the lake, and that was actually a major source of water to the watershed. But because we've drawn down the aquifer for our drinking water, now the lake pushes water into the groundwater. But the cyanotoxins are so labile, so it's transient, they get broken down by bacteria, that they would never make it into the groundwater. They get recycled by the natural bacteria before they ever do. But there's other things that could get transported into the groundwater. Other chemicals.
We are at time. I want to take one more opportunity to thank Dr. Trina McMahon for shameless plug while we have you all in the room. Lakeforecast.org. Go to that website, put it on the home screen on your phone. We have 70 monitors around all 5 lakes who check the lake at least once a week, and if they notice a cyanobacteria bloom, they will continue to check it, and it will be monitored. It will be noted right on lakeforecast.org with a little exclamation point over a green bubble. These monitors, our monitors, were the first ones to detect these large blooms that we've had. So at 7:08, the 2 blooms ago, or this big one, we were already reporting it on Lake Mendota. So if you want to know it right away, we have citizen scientists who are trained to spot these things. So
Physicists and scientists in the room.
Do we have monitors in the room? Look at these people. Thank you very much. You guys are the... So it's just a great tool, and it's a great way. And you know, we partner with public health, and Christy helps us a lot. We'll send people out after if our monitors spot blooms. She'll send people from the lab out to confirm it, and so everything. You know, lakeforecast.org. Okay, next month we are going to have Kel DeWitt talk about Wabeesa wetlands, and we'll be back here at the Edgewater. Hope you can all join us. Obviously, wetlands important for filtering runoff, which causes blooms, so it'll be an interesting topic. On your way out, we have interns and Clean Lakes Alliance staff at every door. If you haven't gotten a parking voucher, they will give you a parking voucher to get out of the ramp for free. Otherwise, have a great day, and get out there and enjoy the lakes.